Welcome to Chapter 6. This is going to be the start of Unit 2. So Unit 1 was kind of an intro into microbiology, whereas Unit 2 is sort of the how and why and a little bit of chemistry when it comes to microbes. So a lot of the function and physiology, if you want to think of it that way. In Chapter 6, we are going to talk about microbial growth, which is going to include nutrition of microbes, movement of molecules in and out of the cell, as well as partnerships and population growth. So let's go ahead and get started on Chapter 6. Let me preface this start of Chapter 6 with Chapter 6 is a lot of definitions. So making flashcards is actually really helpful, but I would start right after you watch this lecture to help you remember those terms. So let's jump in. What do microbes need to grow? Just like all living organisms, we need certain elements to function, to act as our nutrition. So these might include carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, sulfur, and the list kind of goes on. There are certain elements that we cannot make in our cells that we need to obtain from an external environment. So let's talk a little bit more about that. There are essential nutrients that we need to talk about, and we're going to talk about on this slide how much. That is specifically the goal of this slide. We're going to talk about them in terms of macronutrients and micronutrients. So again, these are going to be elements that the cell cannot make on its own, so it has to be found somewhere else. And specifically, how much do we need? So with macro meaning big, think of needing a lot of these elements like carbon, hydrogen, for our cells specifically oxygen. For micro elements, these are going to be known as trace elements, and from the term micro, we know that means little. This means that you need just a smaller amount of these elements, such as magnesium, zinc, nickel. A good example for humans is iron. We don't need a whole lot of iron in our diets, but you can become iron deficient if you do not have enough iron in your diet. So we can also think of what kind of molecule we're looking at as far as our nutrition and microbial nutrition. So this is just another category. If that last slide was how much, this is what kind. And we can classify these kinds in a chemistry distinction versus a biology distinction. Now, the biology distinction is going to make chemists kind of roll over and be like, oh my God, no. When it comes to the chemistry distinction, we have organic versus inorganic molecules, and this is going to be whether or not it contains carbon. So if that molecule contains carbon, it's going to be organic. If it does not contain carbon, that means it's inorganic. When we look at the biology definition of these terms, what we can see is whether or not something came from a living organism or from a non-living product. So the way you can kind of think about this is, if you've ever seen a sci-fi movie or show where they call organisms on Earth carbon-based life forms, that is because we are talking about these organic compounds, okay? And that means it contains carbon, therefore it is a living organism. Inorganic compounds or molecules will not contain carbon and can be things like metals and salts and gases and water. So those are the inorganic, the non-living things. So we've got those two kind of definitions of organic versus inorganic, kind of a chemistry versus biology definition. It should come as no surprise to you that most living things, including microbes, are going to be made up of water. Most organisms on Earth are going to be mainly water, as well as some proteins, some organic compounds. But then I want you to highlight and make sure you know that there are going to be 6 elements that are going to be found most commonly in all living things. So let's talk about these 6 elements more specifically. For these 6 essential nutrients that almost all living organisms share, I need you to know the 6 as well as why they are important, why it is needed, what we need them for in our cells. So we've got carbon first. So this is going to be used for carbs, lipids, proteins, or nucleic acids. And remember, our nucleic acids are going to include things like our DNA and RNA. We have hydrogen next, which is going to help form bonds. Bonds are the important part as well as that source of energy in respiration. So I want you to think of ATP as far as energy goes. The third essential nutrient is going to be sulfur. We use these for vitamins and amino acids. So kind of the building blocks of proteins, if you remember that. Next is nitrogen. This is going to be used for proteins and nucleic acids. Again, DNA and RNA. Phosphate is going to be our 5th element. That is going to build nucleic acids and coenzymes, which we can just kind of think in our head as just enzymes for the moment, as well as help create the cell membrane. And then oxygen. This is going to be used for structural and functional enzymes. So this is not going to be what you initially think of oxygen being needed for, because we need it for respiration. But not all microbes need oxygen for that purpose. There are other important nutrients and elements, but remember that these are the trace elements and not those essential 6. You do not need to memorize these in any capacity. They are just an example of it's not just the 6. We also have more that are needed, but not in the same capacity as we need those essential 6. Now, I know this question has been burning a hole in your head. What do microbes even eat? What could they possibly be ingesting? What microbes eat is going to depend on 2 separate things. It's going to be energy and carbon sources for those microbes. So make sure you kind of highlight and underline that. What microbes eat is going to depend on how they obtain energy as well as what their carbon sources are. So where is that energy and carbon coming from? Let's start with carbon. So how a microbe or organism, because we're not just talking about microbes here, we can also talk about higher life forms as well. How organisms obtain carbon comes in 2 flavors, all right? There are 2 options. The first is known as an autotroph. Autotrophs are going to be known as self-feeders. They are going to essentially suck carbon out of the air, which is kind of cool, versus a heterotroph, which is going to take carbon from something else, meaning they have to consume something in order to obtain carbon. So you can kind of think of a heterotroph as being like us, because we have to consume a pizza or a salad or tacos or whatever in order to intake that carbon, versus an autotroph, which is going to pull it out of the air. You can think of like a plant or algae or phytoplankton. Let's talk about energy sources. So we talked about how you can obtain carbon and what those terms are called. Let's talk about energy. Energy also comes in two flavors as far as how it is taken into the cell or into the organism. You have phototrophs, which are going to be organisms that photosynthesize. You can kind of think of plants, but it's not always plants. And we're talking about microbiology, so we're talking about maybe algae or cyanobacteria or bacteria that is able to photosynthesize, so creating energy from sunlight, versus a chemotroph, which is going to be like us, where you have to, again, eat another organism in order to get that energy. So when it comes to those terms, autotroph versus heterotroph, what we're talking about is carbon. When we talk about phototrophs versus chemotrophs, we're talking about energy. Now, all of that's super fantastic, but we have to combine these terms because we need carbon and energy. You can't have one without the other. There is no organism that needs one versus the other. So what we're going to do is kind of smash together those four terms that we were just talking about: autotroph, heterotroph, phototroph, and chemotroph. When we smush them together, we are talking about organisms and how they obtain energy and carbon, but we're talking about it at the same time. Let's start with the autotrophs. So this is that first type of carbon source type of organism. These are the self-feeders as far as carbon goes, but we're going to add that energy aspect into this. So let's talk about it. The first is a photoautotroph. So these guys can be known as the true self-feeders. They're going to suck carbon out of the air, as well as they are able to photosynthesize light into energy. So they are those true self-feeders. They don't need to intake anything into their cells, into them as an organism. They can just kind of create energy and suck carbon out of the air. Then we have chemoautotrophs. These guys are going to be the ones that are able to suck carbon out of the air, but they still have to consume other organisms in order to get energy. So we have two types: the chemoorganic autotrophs and lithotrophs. Chemoorganic autotrophs are going to be able to suck carbon out of the environment, out of the air, if you want to think of it that way, but then they have to consume organic compounds, living organisms, for their energy. Lithoautotrophs are going to be the weird ones. They are able to suck carbon out of the air, but they consume inorganic compounds. An example that's not a great example, but it's an example I can give you, are the microbes that eat oil. So they are eating that inorganic compound, which is oil, and they are also able to suck carbon out of the air. So that is their carbon source and energy source. That is the two things you have to think of with these terms smushing together. Moving into the heterotrophs. The heterotrophs, remember, are going to take carbon from something else. And then we have to talk about how they get their energy. So we know that they have to consume something in order to get their carbon, but how do they get their energy? Well, the first makes sense. It's a photoheterotroph. We are combining the carbon and energy sources. So energy-wise, these organisms are going to use sunlight as energy. They can photosynthesize, but they need to consume living material in order to get the carbon. A good example we can think of right off the top of my head is a Venus flytrap. So it is a plant that is carnivorous. It consumes living organisms such as flies. Some plants, like pitcher plants, they are able to consume larger things like birds and mice and lizards, but that is their carbon source. But they can still photosynthesize, and that is how they create their energy, if you want to think of it that way. Chemoheterotrophs are going to kind of be, if you want to think of it this way, they are just consumers in both aspects. They have to consume organisms in order to get carbon. That is the heterotroph side. And then chemotroph is going to mean that they also have to consume organisms in order to get their energy. So they are consuming on both sides in order to get carbon and energy. This is like us, right? So we have two types that I want you to know. There's more than just these two types, but there's only two types I need you to know. We have saprobes, also known as decomposers or saprophytes. You can think of fungi. Fungi are a good example of these, as they do not photosynthesize because they are not plants. Fungi are going to decompose living material that has potentially died, like detritus. And that is how they get their carbon as well as their energy from dead organisms. Next is parasites. We are going to talk more about parasites eventually. Remember our tapeworm buddies, our flukes, those little guys. These are going to get nutrients from the cells or tissues of a living host. So versus a saprobe, which is going to get its carbon and energy from a dead organism, parasites need a living organism to live inside or on. Also, parasites, as you can imagine, are going to cause many diseases. Okay, we are going to start talking about how cells can move molecules as well as particles across the membrane. So if you were confused at all about heterotroph versus autotroph versus chemotroph and phototroph, I would highly recommend going back and rewatching that as many times as it takes, because we are going to move right along. So let's talk about how cells are able to move items across the plasma membrane. So there are some mechanisms for how microbes are able to bring nutrients into the cell. So kind of answering the question of how microbes eat. So regardless of whether a cell is going to have a cell wall, they all have a cytoplasmic membrane. And we need to talk about those different mechanisms for how molecules move into the cell. So the first is that it's driven by atomic movement. I want you to think back to chapter, I want to say, maybe 2 or 3, earlier in Unit 1, definitely, when we talked about motility and Brownian movement. Remember Brownian movement? That was movement of a cell, but not motility. So it happens by accident. Atomic movement is precisely what Brownian movement is, where atoms are naturally moving and pushing things. And what can happen is molecules or nutrients can just get shoved through the plasma membrane of a cell. So it's not really active, it is an accident. So atomic movement, think of Brownian movement, it's an accident of movement, but it's not motility and it's not an active conscious choice. Versus molecule movement, which is going to be purposeful. I want you to make sure you know that. This is a purposeful movement. And then we're going to talk about this term diffusion. So remember, when it comes to diffusion, that we are going from an area of high concentration to an area of low concentration. So this is going to be the natural movement of many things. We are going to talk about osmosis first, and move from there. That's a little pun, moving. We're talking about movement of molecules. Gentle reminder of what diffusion can look like. kind of dye in water. It is the concentration of dye is highest in one area where it is being dropped into the water and it's diffusing until you reach a term called equilibrium. This is very important. You can also think of like how a candle will diffuse those scent molecules once it's lit. So do keep in mind that molecules are always trying to reach this equilibrium at all times. You can also think of being in an elevator. If the elevator is particularly full, and then a couple people get off on one floor, everyone's not going to stay in one corner where they were all huddled up together. They're going to disperse evenly within the elevator in order to not be so cramped together. That is diffusion, just with people, which is kind of fun to think about. Okay, so we are on to osmosis. This is going to be the movement specifically of, make sure you circle it, highlight it, whatever you need to do, water. So this is going to be the movement of water across the membrane. Now remember that the membrane of our cells are going to be, this term, selectively permeable. The purpose of the cell's membrane has a very important job, and that's to keep things in and keep other things out. Unfortunately, water doesn't really adhere to that rule very well, and it's just going to move where it wants to, and especially where it's able to move. And that's going to be either in or out of the cell. Now, no matter where it's moving to, water is going to move in such a way in order to reach a balance, which I mentioned on that last slide, which is called equilibrium. The cell is unable to stop water moving in and out. So let's talk about what happens and how it happens. So in talking about osmosis, we do need to talk about a relationship between the cells and then the solution that they are found in. So essentially their environment. We need to talk about something called a solute. So solutes you can think of as stuff being in the water. This can be salt, sugar, proteins, all kinds of things. So what we're talking about with this relationship is called tonicity. Tonicity is going to be that relationship between what the water contains inside of the cell versus what the water contains outside of the cell. Essentially, you can think of how much stuff is in the water on either side of the membrane. So we need to talk about those three possibilities, because there are only three possibilities of this relationship with this stuff in water in and out of the cell. So the three possibilities, those three relationships we can think of as far as tonicity goes, is hypertonic, isotonic, and hypotonic. So hypertonic is going to be where a solution with more solute and less water compared to the other solution. And hypotonic is going to be the opposite. It's a solution with less solutes and more water. So hyper meaning a lot of whatever that stuff is in the water, sugar, salt, protein, whatever. Hypotonic meaning there is less solute, more water. So that is that relationship. Isotonic is going to be an equal amount of solute and water compared to the other solution. Now, seeing these definitions on the screen is probably like, okay, and well, I'm going to give you a visualization here in just a second. You do not have a slide. I would recommend drawing out what I have on my screen to help yourself understand this tonicity relationship. Because when we look at these relationships, we have to look at inside the cell, we have to look outside the cell, because we are looking at two solutions. All right, we have to look at how much stuff is in the water in the cell versus how much stuff is in the water outside of the cell. So let's go ahead and do that. I have three cells here for you, and just know that they have their own solution on the inside and their own solution on the outside. Let's start with cell number 1 and talk about what's going on inside that cell and outside that cell. For cell number 1, we have an equal situation where there's a 25% salt versus 75% water solution inside of the cell as well as outside the cell. So we can see those solutes, so the stuff inside the cell, those little particles, and then outside the cell are equal. What this tells us is that water is going to move in and out of the cell equally because water is always going to move. So this is our isotonic movement of water. So this is the only time where the inside and outside the cell are equal. Water is moving equally into the cell, equally out of the cell. We have isotonic equilibrium. So water's still moving because it has to, and it is equal movement. This is a preferred condition for our cells. The other conditions are going to be less favorable and are what happens with dehydration and overhydration. So let's talk about them. For the second cell, we have a 5% sugar versus 95% water solution inside of the cell. And then it doesn't have to be equal on the outside. It doesn't have to perfectly match those numbers. So 75% sugar outside versus 25% water. That is our ratio of the solution on the outside of the cell. There is more water in the cell than there is outside of the cell. This is a very important thing to make a note of. We also have less solute or stuff inside of the cell. There is more solutes outside of the cell. This means that water is going to move out of the cell because water is going to move where there is less water. Make sure you know that. Water is going to move where there is less water. So that 95 to 5% sugar water solution is going to be pushing the water out because water's moving in the direction of going from high concentration to low concentration. Keep that in mind. Now, the thing we have to talk about here, because we have an unequal solution situation between the inside and outside of the cell, is we have to talk about what those solutions are called. Inside of the cell is a hypotonic solution, meaning that there is more water than there is stuff or solutes. Outside of the cell, it is a hypertonic solution, meaning that there is more stuff, less water. And again, what's going to happen here is water is going to leave the cell because there is less stuff in the cell than outside of the cell. And remember, with diffusion, we are moving from an area of high concentration to low concentration. So water is going to move out of the cell. What this can cause the cell to do is shrink and shrivel. This is not a favorable situation in our cells because this is what happens with dehydration. The final cell that we want to talk about is going to be the reverse of that second cell, where we have a 75%, let's say, protein versus 25% water solution inside of the cell. So we have a lot of solutes, a lot of stuff inside of the cell versus outside the cell, which is a 5% protein, 90% water. So a lot of water is outside of the cell. What do we think is going to happen? Naturally, water is going to go where water is less. So that water is going to go rushing into the cell because it is less water inside of the cell and more solute. The solutes on the outside of the cell, there are less solutes, more water on the outside. Water is going to move into the cell. Because there are more solutes and less water in the cell, this is a hypertonic solution. The outside of the cell is mostly water with a little bit of solutes. This is a hypotonic solution. And then, of course, water is moving into the cell because there is less stuff outside the cell than inside. So That is our final situation there. I will say this, guys, you need to make sure that you know for the exam, if I give you the information that the cell has whatever percent of a solute, and then the environment has whatever percent of a solute, I need you to tell me, is it hypotonic, hypertonic, isotonic? What's happening in the cell? What direction is water moving? And then what happens to the cell? So for our first cell there, remember that is an isotonic solution in and out of the cell. Water is moving, but it is balanced. For the second cell, we have a hypertonic solution outside because there's more solutes, less water. And then inside the cell, we have a hypotonic solution where there's more water, less solutes. That means water is going to move out of the cell, meaning the cell is going to shrivel. For that third situation, we have more solutes and less water inside the cell, making it hypertonic. And then outside the cell is more water, less solutes, making it hypotonic. The water is going to move into the cell, causing the cell to swell. So it is being filled with water, it's swelling. So again, for the exam, make sure you know if I give you the percent of the solute, so salt, sugar, protein, whatever it is, that you should know that it is additive, right? And then whatever is left to get to 100 is going to be that amount of water. So you should know whether or not you're looking at a hypertonic or hypotonic solution based on the percentages. And of course, if you ever have any questions, don't hesitate to ask. This is just a nice review of what's happening to the cell in those different solutions. So in an isotonic solution, again, water's moving in and out of the cell equally. In a hypotonic solution, water is going to move into the cell, causing the cell to swell. In a hypertonic solution, the cell is going to be losing water. Water is going to leave the cell, causing the cell to shrivel or shrink. All right, there are two different types of transport when it comes to moving things across the membrane. And you probably remember these terms from a previous biology course or, or not. That's okay too. We are here to learn. So we have passive transport and active transport. And the big thing you need to remember when it comes to these two types of transport is that passive requires no energy. Active means you have to use energy to move molecules. So let's talk about passive transport first, because there are two types of passive transport you need to know. There's simple diffusion and facilitated diffusion. Simple diffusion is, as the name implies, simply diffusing through the plasma membrane. We are going from an area of high concentration to an area of low concentration. You are just moving down that diffusion gradient in order for molecules to pass directly through the plasma membrane. With facilitated diffusion, essentially the same thing is happening, except you need a buddy. You need something to facilitate the movement of those molecules through the plasma membrane. This is not energy, though. This is going to be a channel or a pore in the membrane wall itself. What this does is create, and I'd write this word down, a bridge. Facilitated diffusion is like a bridge through the plasma membrane in order to move molecules from an area of high concentration to low concentration. Those are the two passive transport types. When it comes to active transport, I don't need you to know any type. There are types, but you don't need to know them. But we need energy in the form of ATP, and it is because With active transport, you're moving against the gradient, meaning you're going from an area of low concentration to an area of high concentration. We'll talk about where this is important when we get to cell respiration, but essentially just know you're kind of moving against that process of diffusion with active transport. Okay, and of course, a lovely visual to help us know what we're looking at. So passive transport, we've got our simple diffusion, where those molecules are able to just pass right through the plasma membrane, going from an area of high concentration to low concentration. We have facilitated diffusion, still going from an area of high concentration to low concentration, but we have a buddy. We've got that bridge, that channel or pore in the plasma membrane that is allowing the molecules to pass through instead of directly through the plasma membrane. And then last but not least, we have our active transport. We have ATP being used in order to move molecules from an area of low concentration to high concentration. So those are those three important processes of transport across the plasma membrane. Okay, so how do we get big things, big particles in and out of the cell? What's important to remember is direction. What direction are we moving? Exocytosis is going to be exiting the cell, so sending things out of the cell, whereas endocytosis is going to be into the cell, moving particles or molecules in mass into the cell. If you remember from chapter 5, we talked about this when it comes to viruses, how viruses use endocytosis as a sneaky beaky way of getting into a cell. Endocytosis can come in two different methods, and it has to do with whether or not you're moving a particle that is a solid or a liquid. So phagocytosis is going to be a type of endocytosis where the cell is essentially ingesting or wrapping itself around a solid particle and then pulling that thing into the cell. That is phagocytosis. You can think of it as the cell eating versus pinocytosis, which is going to be the plasma membrane invaginating, so creating like a pocket or a bubble, and then pulling a liquid in, kind of like sucking on a straw, because pinocytosis is going to be bringing liquids in. You can think of it like cell drinking. So do look at that imagery down there. Phagocytosis, the plasma membrane is kind of extending out and wrapping around the particle, whereas pinocytosis, the cell is invaginating and creating a pocket, sucking that fluid into the cell. There are different things that can cause an influence on how microbes are able to grow. So let's talk about them. So what affects how well a microbe is able to grow? We can break this down into a few different environmental factors, such as temperature, gases, pH, so whether or not the environment is acidic or basic, osmotic pressure. Remember, we were just talking about osmosis. That's going to have to do with water. And then radiation and hydrostatic or atmospheric pressure. That one will be a little bit less interesting, but we'll still talk about it, as well as other organisms in the environment can inhibit the growth of microbes, which makes sense. We'll talk about it. Let's start with temperature. When it comes to temperature requirements, these are the three ranges I care about for you to know. So we have our minimum growth temperature. This is going to be the lowest temperature that an organism is able to survive. The maximum growth temperature, that is the opposite, the highest temperature that an organism can survive. And then of course it makes sense that there is an optimum growth temperature. This is going to be like that sweet spot where organisms survive and grow best at this temperature. What I want you to kind of think about and associate this with is us, humans. We can survive at all kinds of temperatures. We can live in the Antarctic, I mean, not naked and without some type of enclosure to live in, of course, but we can live there. We can live in a desert, also need to have a couple things, but we can survive in those environments. But there is an optimal area of temperature that we prefer to be at. I don't know about you. My household is always set around 70-ish degrees. That is a comfortable temperature to survive at for humans. We do have an optimum growth temperature, if you want to think of it that way, but like it is that most uh, survivable temperature that we are able to live at. But there are minimum and maximums where we can still live, we can still survive. It's just not great. I do not want you to remember the temperature ranges. However, I need you to remember the terms and what they mean. So we have psychrophiles. These are going to be extreme cold-loving microbes. You can find them in the deep ocean, Arctic and Antarctic. Psychrotrophs, which are going to be cold tolerant, but not the ones that are like crazy about the cold, but they can tolerate the cold. These are going to be found in rivers and lakes. Mesophiles, that is that middle temperature. So we fall in this range. Soil, surface, water, most organisms. And when I say we fall in this range, I mean they like to live inside of us. We are within that range that mesophiles are able to survive at. They are the middle loving temperatures, microbes. Thermophiles are going to be heat loving, but not the extreme heat. They can live in topsoil, silage, and compost piles because that is going to produce a lot of heat. And then hyperthermophiles. These are going to be the extremely heat loving microbes where you're going to find these organisms near volcano vents or like hydrothermal vents in the ocean, as well as hot springs. There are two gases that we really care about, and that is oxygen and carbon dioxide. These are going to fall under the gas requirements for microbes and how it will affect their growth. So let's talk about oxygen first. So when it comes to oxygen, not all microbes need oxygen like most eukaryotes do. So we need oxygen, we have to have it, but for some microbes, it can be really bad. Oxygen can become toxic. Let's talk about it. So when it comes to oxygen, as it enters cellular respiration, it can be transformed into several products that for some microbes, some organisms, it can be a toxic product. You do not need to remember both the name and the chemical formula, just know one or the other, because both will be on the exam, but together, of course. So the four types of toxic products that can be produced from microbes processing oxygen in cellular respiration are the following: a singlet oxygen. It can be really reactive and cause damage. A superoxide ion, which is also highly reactive. And let's skip the third one for just a second. We have a hydroxyl radical. This is also going to be reactive. And the third that we skipped and we're coming back to is hydrogen peroxide. This should be the one that you know and kind of maybe have in your medicine cabinet. So this is H2O2. And hydrogen peroxide is going to be something that we use to disinfect wounds. But this is a product of the process of cellular respiration. So what happens is bacteria is not able to convert H2O2, and it can be detrimental to certain microbes. So again, remember, not all organisms need oxygen like we do. And even us, we have to convert some of these products in order to make them safe for us as far as our cells go. So I keep hinting at this process of converting that toxic oxygen or bad oxygen into something that is not going to be bad. You can think of it as making oxygen safe to use. So we have superoxide dismutase and catalase. So these are the two processes on how to convert oxygen, bad oxygen, into something that is safe to use. So some microbes will use these processes because we do. Our cells do this, and some bacteria, some other microbes might do this as well. Now, not all of them do, and specifically, not all of them can. Not all microbes have the ability to do this process and make oxygen safe or oxygen products safe for the cell. That leads us into oxygen usage and tolerance patterns in microbes. So we have 5 categories, kind of like how we had the 5 or the different temperature classifications. We now have 5 categories of oxygen use and tolerance. So we have aerobes or obligate aerobes. These guys require oxygen. We are obligate aerobes. We have to have oxygen. Facultative anaerobes will prefer oxygen but do not need it. They prefer it for their growth. Anaerobes or obligate anaerobes will be killed by oxygen. They do not have the ability to process oxygen into something safe, so they will be killed by the oxygen if it is introduced into their cellular system. We have aerotolerant anaerobes, which prefer no oxygen, but these guys don't need oxygen to grow like facultative anaerobes. They do prefer oxygen, but don't need it. These guys are kind of the opposite. So aerotolerant anaerobes prefer no oxygen, but if oxygen is present, they'll still be fine. And then we have microaerophiles. They require low levels of oxygen. And what we can see happening in that image there is the growth of a microbe in different areas of a tube based on their oxygen usage and tolerance. So what's kind of fun is we're actually going to see this in lab using thioglycolate test, and it basically just shows what type of microbe we have in front of us and how they tolerate oxygen. If they grow at the bottom of the tube, then we know that they don't want oxygen. If they grow at the top of the tube, we know they want oxygen. And if they grow somewhere in the middle, in between, we know that they can have oxygen, but maybe don't prefer it. All right, moving to the flip side of our gas requirements. We talked about oxygen, now we are talking about carbon dioxide. Less interesting, these are just organisms that will prefer carbon dioxide or need it in their metabolism. We call these capnophiles. So they grow best at higher concentrations of carbon dioxide, and sometimes it's necessary, or they are able to create environments that will have more carbon dioxide. A really kind of scary but interesting and applicable example is Streptococcus pneumoniae. Oh, words are hard, guys. The bacteria that causes pneumonia. What happens is this bacteria makes it hard to breathe so that your oxygen levels in your lungs are lower and more carbon dioxide builds up. Now it's kind of a sneaky way to do this, but this bacteria does it because it prefers carbon dioxide. So it's essentially kind of making it harder for you to breathe because it wants it that way. It wants the carbon dioxide to build up. It doesn't want as much oxygen. But that is what a lot of these capnophiles will do. Moving on to pH. So remember that this is going to be a scale. where closer to 1, you are getting more acidic. Closer to 14, you are getting more basic. For organisms that are able to grow and prefer to grow in the middle, around pH 6 to 8, these are going to be neutrophiles. So that is that neutral zone, right? Then when you have acidophiles, these guys are going to grow lower than 5. And then alkaphiles. These are going to grow higher than 9. So they are going to be organisms that prefer alkaline or basic conditions. From there, we have osmotic pressures. Remember that this is going to be the water pressure in and out of the cell. So you have osmophiles. These are microbes that prefer hypertonic environments where there is more solutes outside of the cell. And halophiles, these are a specific type of osmophile that want a high, specifically salt concentration. We talked about this term actually before. You have obligate halophiles, which are salt lovers, and facultative halophiles. These are salt-tolerant microbes. A little different, but still kind of in the same vein as far as pressures go. We want to talk about radiation as well as atmospheric pressure, but we can talk about these kind of collectively together. So when it comes to radiation, these include microbes that use visible light rays as an energy source. So they are a type of phototroph. But most microbes will be damaged by light rays. So not dissimilar to our cells, which these UV rays can cause skin cancer. Now moving on to pressure, we want to talk about barophiles. These are going to be microbes or organisms that love extreme pressures. And usually grow in environments that have a lot of pressure. I want you to think like the deep ocean. The example I have for you is not a microorganism, but instead the very and ever so attractive blobfish. Poor fella, when he's on the surface and experiencing atmospheric pressure, so the normal pressure that we usually have, he looks like that bottom picture. He kind of looks like a deflated balloon with a face. But at his normal deep sea pressure, that's what he normally looks like, that picture on top of kind of the less attractive fella there. Because they prefer that pressure. It's what keeps their cells as well as their organs intact. When you bring these organisms to atmospheric pressure, they will essentially implode or deflate. It is not something that they can tolerate. Most organisms will prefer atmospheric pressure. Barophiles are an exception. The last factor within the growth of microbes has to do with partnerships or relationships between microbes and other organisms. So you can have symbiotic or non-symbiotic relationships. Symbiotic is going to be organisms that live in close, usually mutualistic, but not always, relationships. And it is required by one or both members. So we have mutualism, commensalism, and parasitism. Those are types of symbiotic relationships. Mutualism is where both members will benefit in this symbiotic relationship, where commensalism, only one member is benefiting from the relationship while the other one is not harmed, but is not benefiting from the relationship. And then parasitism is going to be a type of predation where you have one organism benefiting and the other one is being harmed. Usually it is a host because we are talking about a parasite. Now, we do have non-symbiotic relationships where organisms are free living and the relationship is not required for survival. We have synergism and antagonism. Synergism is where members cooperate and share nutrients. It's kind of a like a kindergarten, sharing is caring situation, and antagonism where some members are inhibited or destroyed by others. So it's kind of a make love, not war situation in a non-symbiotic relationship. Now, I very loosely on that last slide already talked about symbiosis. So let's talk a little bit more. So symbiosis, again, is sort of this general term that is going to say that there's a relationship, or you can think of it as a partnership, however you want to think of it. There is some type of one organism interacting with another organism happening. So what we call these members are symbionts. They are the members of the symbiosis relationship. There are three types of symbiosis. We talked about them before. Mutualism, where both parties are going to benefit. So an example is the microbes in our intestines. What they do is help us digest products that we wouldn't normally be able to digest on our own. We have a mutual relationship with the bacteria and other microbes in our intestines in that they help us and we help them by giving them a place to live and also food to eat. Commensalism is going to be a partnership where one organism is going to benefit and then the other partner is just kind of there. They're not benefiting, but they're not being harmed. Kind of an example is the microbes on our skin. Not a terribly great example because we do get benefits from the microbes on our skin, but they definitely do. But we don't always get benefits from the microbes on our skin, and sometimes it's not a good relationship either. If you think of like MRSA, not a good time. Parasitism is going to be where one party, the predator, is going to benefit from the relationship while the host, the other party, is going to be harmed in the relationship. An example is pathogens. We can think of those protozoans that cause malaria. So that is an example of parasitism. Now we can have another type of relationship which is non-symbiotic. One of those types of non-symbiotic relationships is synergism. This is a relationship or kind of a non-relationship between two organisms that benefit them, but it's not necessary for survival. So remember that mutualism, those organisms do need each other, but in synergism, they can form a relationship, but it's not necessary. So together the participants are going to cooperate to produce a result that they couldn't do alone, but they don't necessarily have to do. An example that we've already talked about is biofilms. So this can be things that cause gum disease or maybe some bloodstream infections that involve mixed infections of bacteria interacting together through synergism. So those bacteria are able to create this thing, a biofilm, because they're working together, but they don't have to work together to survive. And then last but not least, we have a non-symbiotic relationship, which is antagonism. So this is going to be kind of a war situation where this kind of non-relationship is going to be a negative one. So it's where some species essentially compete and kill off other species of microbes or organisms, it doesn't have to just be microbes, in order to obtain resources. That's always going to be the kind of end result here, is needing those resources. What we call this process is antibiosis. This is the production of inhibitory compounds. You can think of antibiotics within an environment that end up destroying microbes in that habitat. This is produced by other microbes or bacteria in order to compete and kill off microbes that are taking up resources. which is kind of cool, kind of terrifying, but also a neat area where we are able to obtain antibiotics through bacteria that go through this process. And with that, guys, that is the end of the chapter 6 part 1 video. If you have any questions at all, please don't hesitate to reach out, email me. And again, this chapter is very definition heavy. I would start making flashcards now. That would be the smart move. So make good choices, and I will see you soon for chapter 6 part 2.